So, hello to everyone. First, I would like to thank the organizers for the opportunity to present at this excellent conference. My name is Jose Espinosa-Carrasco. I'm working as a bioinformatician at the lab of Cedric Notre Dame at the CRG. And actually, this is the lab where Nexo was originally developed, as we have already mentioned during the previous session. And I'm also a member of the NF core team. And today I will try to convince you that Nexo and NF core could be very helpful in your research. So you have here on this slide several flagship genomic projects like ENCODE, GeneCode, GTEx, the Human Cell Atlas. And all of these projects share one thing, and the thing that they share is that to process the data that they have and to obtain the results that they obtain, they all rely on bioinformatics pipelines. So I would say that bioinformatics pipelines are the cornerstone of modern genomics and even of modern biomedical research. And of course, for developing and running and deploying your pipelines, you need a language. And these languages are workflow management systems. Workflow management systems bring key features like portability, reproducibility, scalability to your pipelines. And on this plot, what I'm showing you are the citations of several of these workflow management systems along the time. And if you take a look on the green share, this is Nexo, and you can see how it has been increasingly gaining moment until last year when it became the most widely used workflow management system. But of course, if you have a language, you can talk it properly or you can talk it in a bad way. And if you want to talk it like politely, you need a standard. And this standard, it's NF Core. So NF Core, it's a standard for developing good practices, Nexo pipelines. And this standard allows you to do things like several groups contributing to the same pipeline at the same time. And this is because by design, NF Core has a lot of testing that allows to make sure that the features that are added by these different groups along the time are all working as expected and are meeting the standard. And another important thing about this is that these make your pipeline immortal, so to say. So the thing is that many times you are working in a group, you get funding, you develop your very nice pipeline, then the funding stopped and this pipeline got abandoned. And this is a pity for both for you and for the whole community. But in this case, you can see how this small RNA-seq pipeline was started by the SC Life Lab. Then other people come into the pipeline, adding some features, and at some point they stop contributing to it. But other groups took over, and the pipeline keep going. So, as I was saying in my last slide, NF Core makes Nexo a special workflow management system. And maybe you don't know what it's NF Core. So NF Core, it's several things. It's a community of users and developers which seek to collect a series of best practices pipelines of Nexo best practices pipelines. And for this, for having these pipelines being portable, reproducible, scalable, we need to set some coding guidelines. And this is the reason why we come up with some guidelines. And this is the standard. And we also provide with some helper tools to use and develop the pipelines. And here on the bottom, I'm showing you how is the size of the current community. So you can see that we have reached more than 10,000 Slack users. And actually, 2 months ago, we reached 12,000 Slack users. Slack is our main communication platform. We use it to discuss on how we develop the pipelines, how you can use the pipelines, and so on and so forth. And this is the current collection of NF Core pipelines. There are 83 pipelines and 41 that are under development, which mean that they have not yet been released. The first stable release has not been done still. And these pipelines need some components. You need some components to make this pipeline up. And these are the modules. So the modules are kind of the treasure of Nextflow. They have grown a lot. They have exploited in the last times. We have currently 1,567 modules, and these modules are wrappers of bioinformatic tools that you can use in any Nextflow pipeline. So it means that you don't need to use these modules in an F4 pipeline. You can use it in any Nextflow pipeline. And all the pipelines and the modules are openly available for the whole community as open source under the MIT license. So maybe you are wondering how you can contribute to NF Core, on what you can contribute to NF Core. So you can contribute to NF Core at any level. You can contribute at the level of pipelines, at the level of sub-workflows. Sub-workflows are just some of these modules chain one after the other to perform a typical bioinformatic task that can be shared among different pipelines. Then you can also contribute to modules. And if you are interested, you can even contribute to tooling. So to these tools that help us to set the standard and make sure that all the pipelines are working and everything is set as it should be. So yes, and how we guarantee that everything is working as expected and everything is reproducible is because, as I mentioned before, so NF Core is doing testing by design. So anything that you are adding to the code base will be tested before it goes in production. Well, you have, of course, so people also reviewing pull requests. So there are some requirements, like for one thing that it's going to development, you need to be approved by one member of the community, and when it goes to real production, you need 2 members of the team approving it, and things like this. So workflow managers and standards are not something static. They have to evolve to accommodate the new needs of the users. And in this sense, our community and so Nextflow and NF Core are a pretty nice ecosystem, because in fact, in this case, what happens is that NF Core is driving the evolution of the language and the standard. And what does this mean is that when we identify some needs, we are developing pipelines, we have new needs, and then what we do is that we modify the standard to accommodate these needs, and also we try to push this into the language so that the language can evolve and meet our needs. A recent development in the NF Core community was the creation of the special interest groups. A little bit more than a year, than a year ago. And until that moment, the pipeline was structured around pipelines. So we have a Slack channel for each of the pipelines, and users go there to ask questions, developers go there to discuss how to develop the pipeline, and they form kind of a community around the pipeline. But what we saw is that many people, in fact, were not using just a one pipeline. They were using several pipelines. So you can be using ERNA-seq, ChIP-seq, ATAC-seq, because, for instance, you are working in a given animal species, or because you are your focus, your research focus is whatever. So that's why we came up with this idea of the special interest group. And the idea is that this special interest group brings together people that are working that have the same scientific focus, so that they can discuss, for instance, how you have to parameterize your pipeline to work in this given animal species, or also which features this pipeline is lacking for your research, for this given research, and you want to try to incorporate to the code base. One interesting thing is that you can adopt NF Core at different levels. So here I will show you several communities, from one that adopted NF Core in less than the final one that made a fully adoption of NF Core. So the Epitome workflow community from Oxford Nanopore Technologies created a series of workflows to process laundries data for several applications. What they did is that they are using our NF Core template to develop their pipelines, but they are using their own modules and their own sub-workflows. Then we have the Darwin Tree of Life. In this case, this community is also developing workflows for this project. They even mirror our website, as you can see here. But in this case, they are using the NF Core modules and the NF Core sub-workflows from our repository. And when they need new modules, or when they have to modify them, they contribute back to the community, which is very nice. And then we have cases like the Eurofund community, where we got into a fully NF Core adoption, as I will present you now. So I was part of Eurofund, and I will explain now how Eurofund adopted NF Core as the standard for its analysis pipelines. So this plot here is just to show you that in 2050, the world population will reach 9.2 persons. And of course, we have to find ways to feed them. And this will only be possible if we find technical ways of doing this to increase production and with genomics. So that's the reason why there are initiatives as the functional annotations of animal genomes, which are trying to annotate the genomes of several common livestock species in order to increase this production, but not only this, doing it in a healthy way, both for human and for the animals, and in a sustainable way. And there is an European chapter of this initiative, that's the Eurofund, which it's an umbrella for all these projects that I'm showing you here. And actually, I was part of BOSC. This is the bovine regulation. It's kind of an encode for cows. And the aim, when we started the project, we were actually providing the bioinformatics sources for analyzing the data in the project in our group. And the idea when we started the project was that the analysis pipelines that we developed or that we used during the project were a long-lasting resource for the community. For this, we have to choose a workflow management system, as I said before, that ticks all these things. But also, we have to respect the initial heterogeneity of participating groups, because there were groups that were not using workflow management systems, but they have beautiful scripts that were performing very well what they wanted to do. So Nexo, for instance, is a very nice workflow manager system to include these scripts. And also another key point was that we wanted to make results ready for cross-project meta-analysis. We wanted to make the results interoperable so that we can compare between the different species. So we came up, we discussed it, and we thought that NF Core actually was the way to go. And here on this Stanky plot, I'm showing you on the right the different groups. And if, for instance, we go to the red part, one of these, the first red arrow, you can see that we have used one pipeline, that it's NF Core SAREC, and that this pipeline belongs to the NF Core repository. So if you take a look on the BOSC part, we have actually used a lot of the NF Core pipelines. But so for instance, NF Core SAREC, we have used it out of the box. We have not done anything, and we use it. But this was not always the case. Because, for instance, for ChIP-seq and ATAC-seq, we needed new features for what we wanted to do during the project. So then we started to implement these features into the pipeline, and eventually we also get involved in the development or in the maintenance of the pipeline, me and our group, and then we help in the porting to the DSL2 syntax of Nextflow. So you can see that this is a very nice thing, because we get something beneficial for us. We can use these pipelines, but also NF Core got our help in maintaining and modifying these pipelines, and now they are more ready for doing animal genomics stuff. All this story is summarized in this paper that will be published soon, and also the latest development of the Nexo and NF Core. So as I said, Slack is the main communication platform of the community. So I encourage you to join it there. We have other platforms. So we have a very beautiful YouTube channel where you can find many talks and trainings. So you can also join it here. And well, here I wanted just to summarize how will be the way that you can approach the community in an incremental way. So normally what you will do is that you will start using pipelines. Then at some point, probably you will have questions. You can go to Slack and then ask these questions. At some point, probably you will find that there is a bug or there is a feature that you think it's worth implementing in the pipeline. So you can open an issue, and maybe you feel brave and you say, okay, let's implement this feature. So you can become a GitHub organization member and contribute to modules or to the pipelines itself. Then maybe if you have been contributed to a pipeline, at some point you can get involved in the development team of this pipeline. And of course, if you have new ideas and you think there are pipelines that are missing, you can propose them in the repository that we have for it. It's NF Core proposals. And you can become a maintainer of a pipeline. And yeah, I just want to wrap up with these upcoming community events. In October, in the last week of October, it will take place, the NF Core Hackathon in Barcelona. So if you want to code with us, you are more than welcome. For those that are not know Nextflow, there is also a training that is organized by Sequera, the spin-off from our lab that it's now maintaining Nextflow, and also Nextflow Summit just the week before. And I just want to thank all the people that was involved in all the projects and all the stuff that I have presented today, and especially my supervisor, Cedric Notre Dame, also Phil Ewells, who is one of the founders of NF Core, my lab, the NF Core members, and especially the core team, and all the communities, and also Evan and Paolo, which are the guys that were working in the group and that now are the owners of the company. So thank you very much. And if you have any questions, just go ahead. Well, I don't know if it's time for questions now or, yeah? Okay, sorry.
Hello, my name is Nilay. Probably my question is not a key question, so please excuse my insufficient knowledge. And thank you very much for your enlightening talk. I would like to ask that, if I remember correctly, you have mentioned that, yes, Nextflow is an open source with MIT license. And I was about to ask you, why you emphasized MIT license? Is it not possible to use another license? Thank you.
Yes, actually, at the beginning, well, NF Core, it's MIT, and Nextflow actually had a different license at the beginning. The key point, I would say, is that I'm not an expert in license, I have to say, but the key point is that it's an open software. Yeah. It's true that you have to be careful because some licenses allow you to do some stuff that others do not. For instance, the MIT, it's more friendly, for instance, within industry because they can also adopt our pipelines. There are some others that, even if they are open, are more restrictive, and then it's more difficult to for instance, work with industry. But yeah.
Any other question? Thank you. So all those workflow, can they all be, do you provide unique identifiers for each of those workflows, and in particular for each individual version of a workflow? So that if I have to annotate the data that has been obtained by a particular version, I can annotate the data with something more than just github.com nfcore/ and then the name of the workflow. Is there a unique identifier for any given version of a workflow?
Yes. So we release, well, the way it works is that we keep working in development for a pipeline, and at some point we decide that there is enough stuff to go for a release. Then we release, and this release gets a tag in GitHub. It also gets a Zenodo DOI. So, and it's free, so you can always use the same version. So yeah. So it's completely reproducible.
Thank you.
Actually, well, just to add something to this, if you are using, for instance, NF Core tools, you can even just provide the -r argument and put the version, and it will run automatically the version that you want.